third year student in Mechanical Department. Continuing with the tradition of covering Nobel laureates' contributions by our community members, let us start this event. I am thrilled to welcome you all today here to discuss the contributions of Nobel laureates who received this prize in 2024. Thank you for joining us here. We are incredibly fortunate to have with us an esteemed panel of professors and scholars, each bringing a unique perspective from their respective fields. They will be sharing their insights on the significance of these achievements and how they impact academia, industry, and society at large. So, without further ado, let us introduce our distinguished speakers. Our first speaker is Professor Sumeet Agarwal. He comes from the Yardi School of Artificial Intelligence. His research spans healthcare, language processing, and social sciences in AI. Today, he will discuss the contribution
team and IIT Delhi in general for the chance to talk about this. I'd never thought that as a AI machine learning person, I would be talking about the physics Nobel Prize, but then everything is AI nowadays, right? So I guess those of us who've been in AI for a while, we benefit from all the hype. So the physics Nobel Prize this time, as you heard, went to a couple of people who were pioneers in machine learning, which is of course the set of techniques that drives a lot of modern AI. And you can see the citation, foundational discoveries and invention that enable machine learning with artificial neural networks in particular, which is also the family of techniques that is today called deep learning. So of course in 7 minutes I can only give a very quick sense of what the core ideas were and why their contributions were deemed important. I should say that there are many others, as always happens almost with the Nobel Prizes, there are many others who have made valuable contributions. So while we should not undermine the contributions of these two people, it's always the case that actually there's a much larger body of work and a lot of precursors to the people who actually get the recognition, right? And in some ways, the Nobel Prizes should always be taken with a grain of salt, I think, because science is a collective endeavor, right? And individual prizes should not be given too much importance, is what I would like to say. Nevertheless, let's use it as an occasion to understand the ideas, and the persons, of course, are people who made contributions to that. So what has happened in modern AI, which is something at some level all of us know what AI is and the kind of impact it is having, but technically what has driven the shift from more classical or traditional AI, which started out in the 1950s, to the kind of AI we see today and the tremendous growth of both scale and power that these systems have, is the shift towards what we might broadly call the machine learning paradigm, right? And as the name suggests, this is the idea that intelligence can be developed in machines by learning from experience, learning from examples. So a simple instance is, if you think of some task like trying to detect faces, right? So this is something we see with DigiYatra, for example, or many other such systems which are trying to detect human faces. The classical approach would have said, well, to endow a machine with such a concept, you have to kind of analyze what that concept consists of, right? So for example, you might say a human face will be an image which contains certain shape properties, color properties. It might, of course, be defined hierarchically in terms of a face contains other concepts which themselves are non-trivial concepts like eyes. So then you would have to have a separate rule, let's say, giving the conditions for an eye. And the hope was that if you could make a sufficiently extensive set of conditional rules like this, which tell you, if such and such and such come together, then you can call this a face. And then you could refine that to faces for particular people and so on. Maybe then, you would be starting to capture something about how human intelligence, human detection of concepts and processing of concepts operates, right? But that approach very quickly ran into limitations. And what people increasingly found is that actually giving an explicit set of rules or conditions does not work beyond the most simple concepts. In fact, if I ask you to tell me what exactly are the rules that define whether something is a human face or not, even though it's something we do very naturally all the time, we would find it hard to give construct a specific set of rules for it, right? So moving from like a rule-based to a pattern-based picture of what intelligence is and what concepts are that we need to understand and process for intelligence, is what machine learning has enabled, right? By basically saying you can take a lot of examples of some concept like a face, and you can have a sort of flexible model which tunes itself to be able to latch on to that concept, right? So mathematically, this is just some kind of function, but it has some learnable parameters that are fitted to that particular pattern, and then they can either reproduce that pattern or they can be used to distinguish that pattern from other patterns, for example, faces versus non-faces, right? So that's the high-level shift that machine learning in general has brought about. And neural network techniques, which are the specific type of machine learning method recognized by the prize, are the dominant machine learning method today, right? So as the name suggests, neural networks at least loosely are inspired by actually trying to capture neurons in the brain. And the early work on this, starting with the McCulloch-Pitts kind of model back in 1940s, was about trying to capture some basic features of the fact that neurons have different levels of activity and that these seem to be driven by connections between neurons, right? So you basically have these synapses and they can be of different strengths, and one neuron tends to activate another more or less strongly, or it can also inhibit. So you can have both types of connections, activatory and inhibitory. And so neural networks broadly are built on the same kind of idea, that you have these simple computational units, which are called neurons as well in this context. Each of those can have a certain activity level. The simplest example would just be ones and zeros. So some are active, some are inactive. They're just like switches, on or off. And then you have connections of different strengths between different neurons. And the thought is that each one can store some basic property or feature of some larger concept. And the idea that underlies the ability of collections of neurons to capture complex patterns is that there is an associationist quality to how different properties come together into a bigger picture. For example, if you have an elliptical thing and it has a couple of eyes here, it will probably also have a nose-shaped thing here and lips here. And so even if you get partial information, some of these things can start to activate the other things, and a sort of cluster of activity in neurons, which are each latching onto a different portion of a bigger concept, can lead to the activation of and the strengthening of that concept in memory. So this idea is essentially what Hopfield was able to formalize into what is called the Hopfield network. So one half of the Nobel Prize essentially went to this work. And the reason it's the physics Nobel Prize is that at least there's an analogy to physical systems in terms of how this model was set up, right? And so this is nicely illustrated in this figure taken from the official Nobel materials. You can think of these models as operating in a kind of energy landscape where the pattern is stored as a sort of low energy state. And so the way that the kind of network we saw on the previous slide maps onto an energy function is by saying that basically you define your energy as a weighted sum of the products of the activations of individual units or neurons in that state. And then, I mean, there's a minus sign because energy normally in physical systems is something that is minimized. But basically what you are trying to do here is maximize certain kinds of correlations between pixels. And so if you are trying to detect a pattern like a J, right? So what the network can do is to store this pattern in a collection of neurons by effectively saying that neurons which correspond to nearby positions which have the same color, for example, right? So two black dots in the J, they will have a positive connection, whereas if they should have opposite colors, for example, at the boundaries of the J letter, then they will have a negative connection. So even if you get an input signal which is kind of noisy, like that J above, by the dynamics of this kind of network, which is the first equation, so there's an update rule where locally each neuron resets itself according to its neighbors, right? So every neuron is trying to kind of align itself with its neighbors, and that is leading to local coherence or local correlations in this kind of system. That is a very important property of real world images, for example, right, which this is able to capture. So you don't just have a random collection of colors, right? You typically have contiguous patches of black and white in any meaningful pattern. So you can think of the network as settling down to a low energy state by essentially minimizing that energy function. And in the weights which lead you to that energy function, to that minima of the energy function or to that energy landscape, you have effectively stored a kind of associative memory, right? So the fascinating thing about this is that a physical analogy has been applied to the much more abstract concept of memory. How do we remember complex patterns? How do we activate patterns from stimuli, right? So that's how the connection has been made from the maths to the semantics. And then let me just quickly give you a sense of also the second half of the prize, which builds on this more basic kind of system towards models that then became the foundation for foundational points for modern deep learning methods. So in the basic sort of Hopfield network, you have units which correspond to some observable properties like different pixels of an image as we saw, and you are learning some pattern of how these should correlate. In the Boltzmann machine kind of model, which is Hinton's core contribution that was cited in the Nobel Prize, you extend this by having some sort of hidden units, and the idea is that you are still trying to learn patterns of activity, but it may be useful or make the model more powerful if you say not everything is directly observable. For instance, you might observe a collection of pixels, but there might be higher level concepts like the eyes and the concept of an ellipse and so on, which it might be useful to explicitly represent in the hidden nodes. So you kind of extend the model to be able to capture those concepts in a latent fashion. And then, just since I'm out of time, the other core thing which Hinton's contribution very importantly feeds into modern deep learning is ways of actually finding those weights or synaptic strengths in such a model via using data to train or to learn the weights in these networks. So an algorithm called backpropagation, which is just briefly depicted here, right, which is a feedback kind of method. So yeah, so this is my last slide. So this is just to say that the kind of impact that these starting points for deep learning have had in our world today spans across things like ChatGPT, things like applications in medical diagnosis, but also scientific analysis. And this is one reason why it got the Physics Nobel Prize, apart from the origins of the ideas, is that these methods are also being used to analyze data from things like the Large Hadron Collider and so on, and hence are being applied in that setting, among many others, including, of course, protein folding, which you'll hear about. So thanks a lot. I'll wind up here. And
thanks, Sumeet. Okay, great. So thank you, Sumeet. In that case, we can move on to the next speaker.
Thank you, Professor Agarwal, for your insightful discussion. Next we have Professor Ritu Kulshreshtha, Head of Department of Biochemical Engineering and Biotechnology. Her research focuses on cancer biology, particularly microRNAs, disease biomarkers, and tumoral hypoxia. Today, she will cover the work of 2024 Nobel Prize winners in medicine, Victor Ambrose and Gary Rufkin. Let's warmly welcome Professor Ritu.
So, good evening, everybody. And I'm very excited to speak about the Nobel Prize winners in the field of physiology or medicine in the year 2024, largely because it's on microRNAs. So in 2009, in December, I made a presentation for faculty. So they got the prize in the field of for discovery of microRNAs and its role in post-transcriptional gene regulation. So I just want to say that while the first microRNA it was discovery gene regulation was almost like by 2000 it was established, but it took so many years to finally, for the Nobel Committee to finally realize its potential and give the award. But largely it could also be that the application potential of this particular microRNAs in the field of medicine, it's largely in the past 10-15 years that it has been realized. So, let me start with their journey, and let me first introduce you to a set of scientists, Brenner, Horwitz and Silston, who got the Nobel Prize in year 2002, and one of them is the mentor of the current Nobel Prize winner, and I'll just go through that. So these are the three scientists, and they got Nobel Prize in understanding that how organs, the development of organs, and also something related to programmed cell death. And the model system they used was this ugly looking, this worm. So this is C. elegans, very complicated name, but let's call it C. elegans. So this is just very, very small, just 1 millimeter long. And let's say in adults, it's like 1 to 1.5 1.5 millimeter long. But the beauty of this particular worm is the fact that there are many organs, like you can see nervous system, gut and muscle, which is also present in the higher organisms. And one beautiful fact is the thing that you imagine a body which is transparent. So what we can see is, so there is defined number of cells, there are 1,000 cells in this particular organism, and you can beautifully see that how these cell lineages, you know, they are developing, how a particular organ is developing, and you can see the beautiful figures here. So you can actually pinpoint that, you know, which cell is giving rise to which cell, where it is going. So it is a transparent body. So it is because of that that many scientists have used it as a model organism to study the higher organisms. And that's why I've written, actually this has been the basis of 4 different Nobel Prize. And it is Sidney Brenner who actually introduced these nematodes, that they can be used as model organisms. Now the middle person is Robert Horwitz, and he got interested in these these organisms, and his interest was to know that how cell lineages they develop, how the development is taking place, and you know where these cells are going in the body and so on. So one way when you want to understand any particular function or you know any particular phenomenon is that you either overexpress a particular gene or you underexpress a gene, and then you see that what is the phenotype. Now one way to alter various genes is that you mutagenize. So what Brenner did, he had a huge collection of these worms which were mutated in different genes, and he had this whole collection. So Horwitz, he got interested in these worms, and his major interest was to see that how these cell lineages are developed, and this has been a question, still many scientists have this question and they want to answer it. So Horwitz, in his lab, these 2 current Nobel Prize winners, they joined as postdoc. So Victor Ambros is one person who joined his lab, and then another is Gary Rufkin. Now let me tell about Victor Ambros. He did his PhD in MIT in the lab of David Baltimore, who also was a Nobel Prize winner. So you imagine, jaise bolte na baap and dada wala. So his PhD mentor, he got a Nobel Prize, David Baltimore, and now his postdoc mentor, Robert Horwitz, he also got the Nobel Prize. Now I think that his students will be, thinking that probably we can get the next prize. And so these 2 people, they joined, and Gary Rufkin also, he joined, and both were very interested in these 2 mutants which have been shown here. So I told you that there was a huge collection of these worms, and they are called as lineage defective. So that's why the lin words comes from. So these are the 2 mutants. On the top is the normal, then we have a lin4 mutant and lin14 mutant. So when these 2 people joined, they were interested to know that what is the phenotype that they were seeing. See, in lin4, it was defective and you see a very elongated morphology, while in lin14, you see a very small morphology. So one thing that occurred to them was that they have a totally opposite phenotype. So with very elegant genetic experiments, Victor Ambros was able to actually show that the product of lin4 gene is actually inhibiting the product of lin14 gene. So during their time in the lab of Robert Horwitz, they also cloned that, okay, lin14 gene is coding for this particular gene. And then they both joined, Ambros joined Harvard University, and Rufkin joined Harvard Medical School, both as assistant professor. And then they continued with the work on these. So what Victor Ambros found was that lin4, which is one of the gene, the mutant, that actually instead of coding for a protein, it was actually coding for a very small microRNA. And this was a very, very surprising discovery, because till now what we know was that DNA, it gives rise to RNA, RNA gives rise to protein, and protein is the building block of the cell. So this particular observation that the product of one gene which has such a strong phenotype is actually coding for such a small thing like 22 nucleotide RNA, and rather it is not coding for any protein. So this was for the first time that we came to know that there exist in the cell certain genes which actually do not code for any protein but just the RNA. So this started the field of non-coding RNA world. And separately in Rufkin's lab, ho gaya? Okay, just 1 minute. So in Rufkin's lab, what they wanted to see was that in Lin14, that what is there in the Lin14 gene which is actually causing its repression by Lin4. So then they came across that, okay, this is the region in the gene which is causing the repression. Then they both exchanged notes, so that's why a phone call is made. And then he said, I got this Lin4 gene, and he said, I know the sequence which is responsible for the repression. And when computationally they brought the two together, they interestingly found that in the Lin14 gene, there are 7 different motifs to which the Lin4 microRNA binds. So this gave the first hint that indeed microRNAs have the potential to bring about repression of proteins. So this one saying that, dekhan mein chote lage and ghav kare gambhir, don't look at me, but this was the first thing that it was so. That Lin4, such a small thing, can actually repress a whole protein. So what we understand from this was that okay, there exists this non-coding RNA field and it has a huge potential of post-transcriptional gene regulation. But then initially it was thought that probably it's very specific for worm and it doesn't exist in other organisms. But 7 years later in 2000, Rufkin actually, he cloned another microRNA, LED7, and then it became clear through computationally, they came to know that LED7 sequence is actually highly conserved across various organisms. And that gave the thing that it's not specific to worms, but actually, in multiple organisms, it does exist and it has a very important potential. I am just so sorry. So, so microRNAs, so what we understand in all that microRNAs, apart from protein coding genes, which had a very, major status, almost like king-like, that we are the ones who are doing the work. But in the cell, we do have these small non-coding RNAs, which have the potential to regulate these proteins. And on the right, you can see such a complex web, because one microRNA can regulate multiple genes, while one gene can also be targeted by multiple microRNAs, and that's how it's such complex networks that exist in our cell, and that is the basis that we are, such advanced multicellular organism, because we have the potential to fine-tune our gene regulation. So till now, this was all basic. I'll quickly say that, what is the application potential. So in our body, we have these microRNAs, which are actually even present in the body fluid. So these are considered as biomarkers. So these various microRNAs were shown to be perturbed in multiple diseases, including cancer. And they are considered that they are much more better biomarker than many other biomarkers, because they are extremely stable in nature. And you imagine detecting any disease just by taking the blood and, measuring the microRNA rather than any protein in the tissue and so on. So they have a huge biomarker potential, as well as you can understand, because they are involved in regulation of various proteins, they also have a therapeutic potential, because if you want to normalize the level of any abnormal protein in the body, what you can do is just overexpress or underexpress these microRNAs. They will go and bind to these proteins and regulate their levels. So they also have a huge therapeutic potential, and it is because of this overall potential that probably a very, very fundamental observation by Ambrose and Rufkin, it was actually, realized by the Nobel Committee that it has a huge potential in the field of medicine as well. So that's all from my side. This was my first slide that I showed in 2009 to our department when I applied for a faculty position, and see the microRNAs were the, basis for it. So thank you all. That many funding agency, while giving money to me, they asked that, Ritu, it can have off-target effects. So these diseases, they are multi-pathway diseases. So imagine one microRNA at the same time, if it can regulate multiple pathways, it will be better than rather, just one specific target that the current therapy options are. So still there is, there is no microRNA drug in the market yet. There are multiple in clinical trials, but you're right that this off-target effect is something that we have to deal with, and there are many, changes they are making in microRNAs for the same.
I have a question, a very quick question on that gene
So CRISPR and Cas actually they remove the gene completely. But where microRNA comes is at the RNA level. So there and so it is at the gene level for the CRISPR-Cas, but this is at that you can modulate the RNA levels and ultimately the protein. So in general, it's called as gene silencing, that the expression of the gene is being affected.
Welcome Professor Tarak Karmakar from our Department of Chemistry. His interdisciplinary research focuses on simulation methods in chemistry, materials, and biology. Professor Karmakar will discuss the 2024 Nobel Prize winners in chemistry, David Baker, Demis Hassabis, and John Jumper, for their groundbreaking work on proteins. Let's give him a warm welcome.
Okay, so good evening, everyone. It's my pleasure to be here on this platform to talk about this Nobel Award, and thanks to Subodh for the opportunity and the entire team for organizing this event. So let me start with this quote that I collected from the Nobel Prize website, that life could not exist without proteins and that we can now predict protein structure. See why they have written this kind of statement. Now, so these three gentlemen, they, David Baker, Demis Hassabis, and John Jumper have been awarded this year's Chemistry Nobel Prize for their remarkable and groundbreaking work on designing some computational tools that can predict or that can be utilized to design proteins, new proteins which do not exist, and you can predict 3D structure completely from sequences, and that's kind of remarkable groundbreaking work. And so this is what these guys have done. All three guys, they started playing some games with some toys, computers, and then out of 20 amino acids, which are the building blocks of, let's say, our proteins, they could get the three-dimensional structure of the protein, which are typically active. Now, those of you who you are those of you are less familiar with biomolecules or biology like me, so just a quick introduction of proteins. Proteins are building blocks of life, more or less, and then starting from transporting gas molecules like oxygens to ion channels, where the ions go from extracellular to intracellular part, and some catalytic reactions that are very useful for your metabolism or whatever chemical reaction that take place inside cell, to giving strength to your muscles, including cells, and stimulating the neurons. You need proteins everywhere, and that's why it says that proteins are building blocks of life. Now, quick history of protein structure, how protein structure evolved. And I quickly go back to 1957, where John Kendrew, for the first time, solved the structure of hemoglobin using X-ray crystallography. And then look at the structure on hand. Before, I don't know whether 3D printer that time came or not, but these guys could build it by themselves and showcase their structure of hemoglobin. And from that time onward, several hundred thousands of protein structures have been solved, and a repository system for protein crystallographic data was created by Cambridge Crystallography Data Center along with this Brookhaven National Lab in collaboration with. And now look at this plot. So as you go beyond 2020, it's kind of grows exponentially, and we have almost like 200,000 protein structures available on this website, which is called Protein Data Bank. And you can log in there and kind of check the latest deposited structure, and you can do some analysis on that. And look at this, it needs almost a couple of hundreds of TB of data. And why I'm saying this thing, because this has some relevance to the Nobel Prize. Now, another thing that I must mention, 1972, another Nobel Prize was awarded for protein folding problem, and that time it was awarded to Christian and Finson. And this guy, what he did, he found out the idea that these guys utilized today or this year. What he did, he did a lot of experimental work and found out that the sequence, the amino acid sequences encode the information of 3D structure of the protein. And typically, especially he has focused on this ribonuclease protein, and what he found is, in summary, the information of the 3D structure of the protein is solely encoded in the protein's primary sequence, that is the string of amino acids. So the proteins are formed like this. You have amino acids which has NH3 plus and COO minus, one unit, and then you couple another one and keep on doing it, you get a linear chain, and eventually these guys fold because of some mysterious interactions which is encoded, and eventually you get this native structure or many of these structures. So that was the thing, that was the idea that was built in 1970s or even before that, right? Then the problem is like this: if you have 100 amino acids protein, right, and each amino acid can have 2 conformation, then in principle it can have 2 raised to the power 100 structures, right, and that corresponds to almost like 10 raised to the power 30 possible structures. And if imagine if one folding event takes, let's say, assume picoseconds, quite fast, you need 30 billion years to sample that configuration space. So there is a paradox which is called Levinthal's paradox, because if you really follow this kind of rule, then probably we may not get this kind of folding states. But then the proteins, they have local templates and local interactions by which the sequence find pathways through which it quickly goes to the folded state. And this is where I am showing one kind of molecular dynamics simulation movie. Maybe with the sound it will be better. So this is a real molecular dynamics simulation trajectory of 1 millisecond done by Vijay Pandey's group, which shows how protein folds starting from the sequence, right? It's very complicated, but look how quickly it folds in 1 millisecond. Then why do we care about structure prediction? Well, we can we can crystallize the protein, we solve it, and then we get the structure. Then what is the need of computer? The need of the computer is here because this whole process, which looks really innocent and very nice, colorful, it takes few years, starting from, let's say, protein sequence to crystallize it, and then you go to the XRD machine, you get the diffraction pattern. Okay, you need a computer here. People used to do before on graph paper, and then you get the structure of the protein, 3D structure of the protein, and eventually what you get this structure. But computers really help. Now that Anfinsen told that from the sequence you can get the structure, then why don't you use computers to do this job? We don't need anything else. This is how the computational, the importance of computational methods came into the picture. And David Baker, 20 years back, he got the idea, he got the code, which he decoded. And with the advancement of artificial intelligence, now the 50 years protein folding problem is solved, right? And earlier days, David Baker, what he did, he started utilizing from the sequence, use some genetic algorithm, some evolutionary templates, along with physics-based model potential, he could get the structure of protein, which is like 93 amino acids protein, and this is computationally designed protein. It didn't exist. Later on they synthesized it and they found out there's a perfect overlap between this predicted and the crystallized protein. Now Professor Sumeet already talked about this artificial intelligence, and it was awarded to these 2 guys, Hoffield and Hinton, for their discovery of utilizing neurons or neuron mimics to do this kind of complex job. And probably you see the neurons, how they are firing the signals to a perceptron, which it has dendrons, you can assume. It processes it, and then it passes the output towards the next neuron. This is how it works. And then you combine all these guys and make it deep neural networks. You have literally a lot of hidden layers, and that can process a lot of complicated stuff and give you some output which is meaningful. Now this one, yeah, probably I'll take 2 minutes. So this whole idea of deep learning AI, there's this guy Demis Hassabis. He used to be the CEO of DeepMind, right? He used to play chess during his earlier days. He used to be like second champion, not the top one. But then he utilized this idea of playing chess to designing some algorithms for predicting proteins, and that's remarkable. And what he does, what they did actually, they have used something called multiple sequence alignment as inputs from different species of sequences, and they have used something called templates, pairwise templates of amino acids. And they pass these inputs to something called transformer, which combine this information to give you some kind of structure, and they iterate it to refine the structures and eventually give you some structures which are pretty close to experiment. And their mean, you look at this RMSD, it's less than 1 angstrom. Can you believe it? So they predicted 200 million structures. PDB has 200,000. Now we have 200 million structures available to us to do science. The impact of AlphaFold, I told you 200 million structures, which are being used by 2 million people across 200 countries. That's the impact of AlphaFold. And these are two examples where they have computationally predicted proteins which are yet to be crystallized and solved, which has medical relevance. So the question is, what is next? Is everything done? Answer is no. This is the beginning. So what we need to do is, starting from now that we have the structures, what we can focus on is the dynamics of the proteins. And this is, look at this is COVID whole virus capsid simulated in coarse-grained model, and to understand dynamics. And then you have small protein, you can see the opening of the enzyme, protein RNA interactions, again related to RNA therapy, which Professor Ritu talked about. So these are the things that the community is moving. Already 2013 Nobel Prize was awarded to some of these guys for developing these tools. So now we are at very interesting time where we can utilize state-of-the-art technologies, computers, very powerful computers, including our IIT Delhi HPC, right, to do this kind of cool stuff, applying DeepMind. So now these guys, look at this. David Baker, they have created 2 games. One is called Fold It, and there's another one called Rosetta. So those of you who like to play games, try it. You fold your protein, and that's how they build the whole science, right? That's the enthusiasm. Few things that probably we need to move, we need to focus in the coming years, which is something like understanding large macromolecular complexes. Predicting one structure is not enough. You need to understand RNA-protein interaction, protein-protein interactions. Then drug discovery, we need to find out tools to kind of find out drugs for any, let's say, target, irrespective of whether it's protein or nucleic acids. That's another area. Climate and environment, you can design proteins following David Baker's tools, or you can design your own tools to design enzymes that can eat plastics, or that can give you some chemicals that can be utilized as medicines, or it can sequester some toxic gases and convert it to useful chemicals. So with this, we are heading towards something very exciting using computation. And this is another video that I collected from Harvard University, Life of the Cell. With this, I thank you all for your kind attention.
Unfortunately, we are well over time, so if you have questions regarding this presentation, I encourage you to catch all the time after the
Thank you, Professor Tarak, for your valuable insights. Our fourth speaker, Professor Abhijit Banerji, is from the Department of Humanities and Social Sciences, specializing in socio-political studies and cultural analysis. He will discuss the work of Darren, Simon Johnson, and James A. Robinson, Nobel Prize winners in economics. Please welcome Professor Abhijit Banerji.
And how do I move it forward? Download कर लेते हैं. Oh, it's already downloaded.
before even it was awarded.
Downloads में होगा. Thanks Subodh and thanks everyone. So I guess I could say friends students and colleagues, I come to praise Acemoglu, Johnson and Robinson for winning the Bank of Sweden Prize in Economic Sciences. As Sumeet was saying, that doesn't mean that others can't be praised, and that also doesn't mean that others can't be, that Acemoglu, Johnson and Robinson can be criticized, but in 7 minutes, let's just praise them, right? So, Acemoglu and Johnson are economists in MIT, and Robinson is at the University of Chicago. My perception is that Robinson is the most serious and Daron Acemoglu is a sort of polymath who does everything. He is not from this planet. He, amongst other things, he works with his spouse who's head of the Department of Electrical Engineering and Computer Science at MIT. So they do a lot of economics of networks, macro, etc., etc. But this prize is given for some common work that they've done. It's not in unsubstantial amount. Maybe there may be 20 papers, of which I've only read a few. So economists often ask what determines prosperity and economic growth. And if you look at the production sector of any economy, there are these inputs like human capital, which is the quality of labor in terms of how educated it is, etc. Physical capital in machines and so forth, land and land quality, technology. And then one can say that, the economy grows as these inputs and as technology grows, right? So those are called proximate determinants of growth and prosperity. And each of them is very complicated and has a literature of its own. But a bunch of other economists asks, possibly deeper questions, but possibly questions not as easy to answer, on what are the fundamental or root causes of growth, which is, what makes one economy, what makes physical capital in one economy grow faster? Why does one economy invest more, save more? Why does one economy succeed better in R&D, etc., etc.? And so Acemoglu, Johnson and Robinson, AJR from now, are in this camp, which is the institutions camp, and they have sort of brought back into popularity the role of good and bad institutions in enabling prosperity or in enabling stagnation of an economy. So what are these good institutions? Without getting into specifics, good institutions are those that empower citizens at large to invest in these factors of production like education, health, to do R&D, to invest in technological change, etc. by basically enabling two things. One is that flows of investments should flow to sectors that have the highest returns, and some economies are better at doing that than others. And the second is that good institutions must be those that protect these investments and returns from the investments. These returns from the investment should not be expropriated by a few or by the state or by others. Whereas bad institutions are those that restrict property rights to an elite, to a small elite, who then may manage to extract rents from citizens at large. So in some sense, you can see there are some similarities with Marxist thought, but at this point of time, I always like to keep ideology apart and just look at what's in front of me. So a bunch of others have proposed other root causes like geography. You might have read this polymath Jared Diamond, who has this famous book called Guns, Germs and Steel, and proposes that geography is a fundamental cause. There are others historically, including a lot of sociologists who look at culture. Then even amongst people who look at institutions, there are others. The last serious work was done by these economic historians, North and Thomas, who thought that institutions are by and large always efficient. then there are others. So what is AJR's X factor? So roughly it's this. I should just add the caveat that economics doesn't work like some of the sciences where the discoveries are very, very sharply defined, like a cure for some cancer, or discovering some causes of cell death, and those are absolutely wonderful. But you must also wonder why, with so much scientific progress, much of the world is still very poor. So that's the focus of people who think about prosperity and institutions. So the first contribution of AJR is that they show, or they purport to show, that institutional quality, the quality of how good an institution is, it causally impacts prosperity. Causality in a well-defined sense. If there are computer science colleagues, then in Judea Pearl's sense, for instance. And in order to do this, the first paper, and a very famous paper, which we'll talk about a little bit, which was called The Colonial Origins of Comparative Development, they used this idea that colonization left basically two kinds of countries or regions and two kinds of institutions. So the relatively good ones and the bad ones, which were extractive in the sense of extracting rents from a large part of the citizenry. So that's one contribution, and that's an empirical contribution working with data. Along with that, they have a bunch of models of social conflict. So they model institutions as equilibria that come out of social conflicts between different groups. So we will not have time to go into this modeling. So and the third, probably equally impactful contribution is that this has spawned a huge subsequent literature and the use of data and sophisticated causal tools to unpack questions of institutions, history, and economics. So just some pictures from this first of their famous papers. So on this axis is protection from the risk of expropriation between 1985 and 1995, done by a firm that looks at country risk. So what is expropriation risk? It's the risk that returns or your investments or your capital will be expropriated by someone, maybe the state, without adequate compensation. Right? So it could be investments of capitalists, but it could also be that land from peasants is taken without adequate compensation. So the bunch of these things would fall in this category. And so if there's less expropriation risk, that's good, and that's the increase on the horizontal axis. On the vertical axis is log GDP in 1995. So on the x-axis is a proxy for good institution quality. And this is showing that better institution quality in terms of protection of investments leads to more prosperity. Right? This is not a causal inference. So, for instance, it could be that the richer nations, the ones with higher GDP, have more resources and they improve institutions. Or there could be a third factor by geography which could influence both. So, how to tease out whether institution quality itself has a causal impact? So, in this particular paper, they use this, it's a very old idea. They use this idea that sometimes you can determine that part of the variation in institutional quality which is exogenous, which is not driven by whether the nation is rich or whether there's a third factor, etc., etc. And to do that, you need a variable which we call an instrument. So they argue in this paper that the disease burden in different economies, in medieval economies, was quite different from each other. And when the colonizing countries went and settled, it was easier to settle in countries with lower disease burdens, lower burdens of malaria, yellow fever, etc. And where they settled, they built better institutions. Where they could not settle for long, they extracted resources and built bad institutions. And then, there is an argument here in this part where they argue that institutions have great amount of persistence. An extractive institution built 500 years ago could last for centuries. And so if you look at GDP in 1995, it would still bear the impact of deep history. Right? So the rest of the slides, let me not explain, but just show them to you because they've been generated. And there's lots more, but we're out of time.
On the second proposition on social property, this kind of sounds like the Hegelian dialectic and the Hegelian class conflict from Marx. Right? So it's very interesting and intriguing to see that the biggest come full circle and is going back to the Paris problem.
I don't do Hegel. Let's discuss this later. So there are some parallels, but I look at it like an economist. So we can talk about it later.
We are pleased to have Dr. Ikeda from Department of Management Studies, specializing in business cycles and macroeconomics. She will discuss the work of the Japanese organization Nihon Hidankyo, forgive me for the pronunciation, Nobel Peace Prize winners in 2024. Let's welcome Dr. Ikeda.
The outreach team and the Professor Sumeet to give me this opportunity to talk about Peace Prize. It probably will be different, slightly different from what it has been discussed, but for me it's very emotional being Japanese to talk about peace and then nuclear weapons. So today I got the opportunity to present a Nobel Peace Prize by the Nihon Hidankyo. In Japanese in full form, it is Nihon Gensuibaku Higaisha Dantai Kyogikai. And it is in English, Japan Confederation of A and H, so this atomic and hydrogen bomb sufferers organizations. So for Japanese, history of war and especially for this nuclear bomb left such a deep scar to this day. And talking about nuclear is very much sensitive, but at the same time with the rise of the climate change concerns, and also the geopolitical concerns, again, the debate of the nuclear is coming. And India is also the nuclear nation. So for this 7 minutes, I hope you can also reflect on what technology would do, what is the consequences. And I'm not judging good or bad, but I want you to think for some time by using this opportunity. For this, I just want to begin by showing this video. Should I just click here? Short clip I wanted to show you is that how the U.S. dropped the atomic bomb to Hiroshima, on Hiroshima. Sorry. that's the value of the US, democracy on all that. So basically, this is something that I also find it very fascinating to understand what is also the ideology and things are coming into when we talk also about the Tokyo trial in coming few slides. So these are a few pictures and some drawings of the people who were affected by these atomic bombs. Of course, I could have put much worse ones, but I just wanted to show it to you, a few pictures and the drawings to understand how severe that was. Actually, the 120,000 people were killed by 2 bombs that was dropped on Hiroshima and Nagasaki. It was 6 August and then 9th August. It was 2 bombs. And have you ever watched Oppenheimer? Yeah, so if you have watched that, you know that the Manhattan Project, that was leading to this precisely. And so the US wanted to drop the nuclear bombs so that, they believed that the war could be over. 15th of August, of course, it's the same day as the Independence Day of India. It's a different year, 1945, that Japan surrendered. So this was actually leading to the surrender of Japan, but the survivors was 650,000 people. So we are talking about these people, and then this organization has been voicing the suffering of these people and advocating for the termination or not use of the nuclear weapons. And if you have ever chance to go to Japan, please go to Hiroshima. This is a building that we always talk about. This is a symbol of the war. So this is called the Genbaku Dome in Japanese, and there's a peace memorial park is also there. So when Japan hosted G7, all the presidents and the head of the states were standing in front of this small fire in the park next to this place, and then they also pledged for the peace and all that. But of course, in reality, it's different things going on at this moment in time. So coming back to the Nihon Hidankyo, so they are awarded for, the motivation is for its efforts to achieve a world free of the nuclear weapons and for demonstrating through witness testimony that nuclear weapons must never be used again. Because this is the organization, so they don't have a specific sort of individuals to be given. But if you see these 5 people, so they are the sort of head of the organization, and the person in the middle, Mr. Tanaka, is leading this organization. He's 92 years old. And as you know that the war ended in 1945, so the people who are surviving are also getting older, and my grandparents fought the war. My parents suffered from poverty and recovery and then prosperity. We are born with all the prosperity without having any issues. But next generation don't know anything about war. So we don't know what is going to come to the next generation. But so as you see, that these organizations are also struggling to tell the causality of the war also. So it's also symbolic to get these prizes so that everybody sort of revisit the history and then to learn from it. And Nihon Hidankyo, what they do is primarily, so they were established in 1956, and then they have 2 main objectives: to promote the social and economic rights of all Hibakusha people, and also to ensure no one ever again is subject to the catastrophe that befell all the Hibakusha. So no more, basically, Hibakusha. That is what it meant. And they have a website, so sorry, I don't have enough time, so if you're interested, please go and have a look what they do. For a few things I just want to mention is actually this is the second Nobel Peace Prize that is given to Japanese. It was given to former Prime Minister Eisaku Sato. It was also related to the war, related to the nuclear. So he was basically, agreeing actually with the US that Japan do not possess or produce nor permit the introduction of nuclear weapons. And if you know that Article 9 of our Constitution prevents to have the war, go to the war, we also don't have army. We have self-defense force, but we don't have army. So this is a big debate at this moment in time because of China, North Korea, Russia, these threats are coming. So these are now, it's a big debate whether we should change for the constitutions or not. But these will lead to certain implications. Of course, Japan is an ally of the US. One thing I also wanted to mention, I hope you know who he is. for Japanese, he is the person when we talk about Indians. He is the judge, Radha Binod Pal. He has been stood up for Japan when the Tokyo trial after the World War II happens for war criminal. So he was basically saying that Japan was forced to getting into the war because of the resources, particularly the oil that was cut by the US, so that Japan was actually in this war context, there was no choice but going to the war. So he was basically stood up in the principle of the law, and he was defended Japan. So we all know who he is. So I just wanted to tell you about it. And then the last slide is this, that what I mentioned, that the park is basically, this is what it is saying. So we don't repeat this disaster or not repeat the evil, but again, the debate over nuclear deterrence and then technology, and then now warfare is also changing. The drones and then others are now being used. So the war and the humans and the lives are now becoming much more detached. Then we don't really understand the consequences of the war and the nuclear use. So I hope that we can sort of reflect on what technology actually have an implication, not only to talk about what technology per se can do. So that's something that I sort of closing with this. Thank you very much. Sorry, I can't hear. Sorry.
Can you hear me now? Yes. Yeah. So I wanted to say that it is a very moving peace prize. I have always a recurring question: why the second bomb was floated? Why the first Hiroshima bomb would have settled the fight? Is that a premeditated? Was there any deliberation why the second bomb was? So the reason I ask is people in this current world already have a plan how many bombs to throw. Is that some bearing that people need to talk about?
So that's something that I also need to study about and then learn about it. To be honest, I don't have an answer to that. But what I know is that the first bomb and second bomb, the type of bomb that was dropped was different. And then they definitely avoided Kyoto and then some other, specific sort of significance to the areas that has the emperors and all that they avoided. So there's also the debate why it was Nagasaki also. So I think if you don't mind, I'll get back to you on the why the second bomb, but they're precise.
Probably it was an opportunity for the scientists to test out their more.
Yeah, so actually Oppenheimer was really keen to actually do that.
We can take this discussion.
Yeah. Thank you. Sorry. Yeah.
Finally, let me introduce Mr. Nikhil Sanjay Rekha Adsule, a senior research fellow in the Department of Humanities at IIT Delhi. He will discuss the work of South Korean author Han Kang, Nobel Prize winner in literature in 2024. Please welcome.
Nobel Prize in the literature. I think you should clap hands for her. So the citation says, for her intense poetic prose that confronts historical traumas and exposes the Thanksgiving committee mentioned. But I would like you to hear some of the words which she has written from her very famous work published in 2016, for which she got Nobel Prize. Earlier it was in 2014. That is nothing but The Vegetarian. So it's for you. It's your body. You can treat it however you please. The only area where you are free to do just as you like. And even that doesn't turn out how you wanted. So this is the first one. And second, what she says. She's a good woman, he thought. The type of woman whose goodness is oppressive. So this is how Han Kang writes, and how she confronts the historical traumas and the fragility of human life. But to understand her writings and to understand the hermeneutics of her writing, we need to see, or we need to locate Han Kang in her historical linearity. So first we need to know that she was born in 1970. And 1970s was the decade when your Thatcherism and Reaganism started, or the so-called Washington Consensus started. One. And her first work comes out in 1993, which is nothing but a post-Soviet world, or we officially say that it was the end of the Cold War. 2. And her works begins with that. She writes with poetry, then she goes on to write short stories, and then she turns to the novels. 3. So when we are looking at her, I will follow 3 frameworks as a scholar. First, the era in which she is writing. So I will borrow the framework of Lyotard, where he says that she is writing in this era of incredibility of meta-narratives. There are too many narratives, and within that meta-narratives, our hand can be right. 1. Second, whenever I read her writings, I find out 2 important things, which again I will borrow the framework of first, Hortense Spears, a psychoanalytical feminist, where she talks about the connection between the soul and the body. So she doesn't bifurcate them, but she talks about their interconnectivity and their symbiotic relationship. And 2, I will borrow the framework of Italian feminist Luisa Muraro, and in her work, The Symbolic Order of Her Mother, she writes that a woman needs to question the phallocentric and phallogocentric discourse. This is what Anne does. She questions those discourses. She questions who is initiating that discourse. And lastly, when I talk and when I see her work in the terms of society, so there is a beautiful article which has been written by Guardian which says that, thanks Sanjay, you documented the so-called Korean idea of democracy. And this idea of democracy is embedded in nation and nationalism. And this nationalism is not, as Benedict Anderson says, an imagined community. But this idea of nation, nationalism, is embodied in the body of the human beings, is embodied in the body of Koreans. And that is being foregrounded. That is being foregrounded as an antithesis. If her work, if you see, she is also working on her 1948 and 1949 novel, which is again what? Protesting against the dictatorship in the Korea. And third, people say that thanks Sanjay, so this is the interview after she won the Nobel Prize, thanks Sanjay, because you brought our trauma, you brought our historical injustices in a tangible form, which is nothing but writing, and now as she has got the Nobel Prize, it has been documented and it has been read worldwide. Thank you. This is what I do. In time. Yeah, thank you.
Presentation, and it was quite enlightening. Thanks everyone for sparing your time liberally for this event. Thank you all the speakers. Without you this event would not have been possible. We hope to continue with this tradition because Nobel Prizes are given each year. So hope to see many of you next year around similar times. All right, thank you.
Hey. नहीं मेरे को याद नहीं है
नहीं ये क्या इम्प्रॉम्प्टू था या पहले से सोचा हुआ था
तो वो उसको नेगेटिवली रेगुलेट करता है
हरियाणा
अच्छा अच्छा
थैंक यू सर गुड जॉब रियली एंजॉयड थैंक यू सर because yes
otherwise वो क्या होता है कि शाम के डिले हो जाते हैं everybody is happy to go back home
but this is fantastic.
we should continue with